Yeah, the lecture today this lecture will focus on effects of arousal in fMRI data. And I'm going to approach this largely from the from the perspective of what kinds of changes are happening in your neuroimaging data as people become more drowsy. And so just to set this up, suppose that you have, oops, is this in place? Okay. Suppose that you have decided to volunteer for your friend's fMRI study, and you arrive, and it's after lunch, and you've been asked to maintain attention to a task. If you've done this before, you might realize that it's actually pretty hard to stay awake the entire time. And especially in scans that maybe don't have a lot of engaging stimuli being presented. So in resting state scans, you're asked to maybe keep your eyes closed and try not to fall asleep or to maintain attention on a fixation cross. And in fact, there's been research into how long it takes for people to fall asleep in a scanner, and it's been found that a number of individuals will start to fall asleep within the first few minutes of being in the fMRI scan. And so, if you think from the viewpoint of a researcher trying to study cognitive neuroscience with fMRI, this would definitely have implications for our studies because the state changes that are going on, drifting to drowsiness, this may impact both the fMRI signal as well as subjects' behavior during the experiment. And so arousal here is going to refer to the spectrum of functional states, including alertness to drowsiness and falling asleep. And in the literature, you may also see terms like vigilance used to refer to the same phenomenon, although in some contexts, vigilance might also refer to a more explicit, attentional component. And arousal is indeed this widespread effect, engaging the brain and the body and your behavior. So as you're drifting between these different states, the characteristics of your neural activity across the brain are changing. There are subcortical systems involved in coordinating arousal changes that project widely throughout the cortex. There are also shifts in the physiology of your body, such as your heart rate and your breathing patterns. And as you may discuss later in this class, changes in your physiology also affect the bold signal. And of course, alertness is also impacting how people perceive and process information, respond to stimuli, and so the behavioral data in your experiments will also reflect your underlying state. And so overall, there are these many different aspects of fMRI experiments could be affected by arousal states, and there are many intertwined effects that contribute. So I think there are several reasons overall why we might want to consider arousal states in our studies. So firstly, if there are differences in arousal either within a scan or between scans or different individuals, then this could potentially be a source of variability in our data and in any of our resulting findings. And this might be especially important when we'd expect some kind of systematic differences to occur, for instance, between patient population and controls, or over time, or if your subjects are on certain medications, and so forth. And so if we can clearly understand the influence of arousal on fMRI, then it might improve how we interpret our data and also boost the sensitivity to specific effects. But importantly, it's not just to be considered a confound. This is actually a really important aspect of how we function, and many neurological or neuropsychiatric disorders actually come with altered arousal regulation. And so if these effects of arousal state are showing up in our data, then we can also consider this an opportunity to use fMRI to study these whole brain circuits involved in coordinating these state changes, as well as how they may be disrupted in disease. So let's now start going into the kinds of fMRI signal and connectivity patterns that may accompany changes in arousal. And so one observation has been that the amplitude of your fMRI signal fluctuations tends to increase as people drift from being more alert to more drowsy and falling asleep. And so this is actually data from an early study where they were scanning somebody for quite a long time. I think they first had some tasks going on, and then they said, okay, just close your eyes. It's okay if you fall asleep. And they noticed that the variation in the bold signal seems to increase, and that presumably this could be linked with the person falling asleep. This has been replicated in a number of other studies as well. So this is a sleep study from WashU. They also found that if you look at the amplitude of these bold fluctuations, that it tended to be higher in sleep compared to wake across multiple functional networks. And these kinds of changes can be observed in shorter resting state scans as well. So not necessarily over an hour of scanning, but so this more recent work took data from the Human Connectome Project and the UK Biobank, and they found that you can observe these trends where certain brain regions are increasing their amplitude over time. Actually, this is quite a widespread global phenomenon, although some brain areas seem to show more prominent changes than others. And those tend to be found in sensory areas, actually. So like primary visual, auditory, and motor cortex are some of the regions that tend to show this phenomenon most strongly. So that's a little bit what's going on with our raw signals. What about measures of functional connectivity, region-to-region correlations? So one general observation has been that across the cortex, correlations between brain regions tend to become more global when people are drowsy and start to fall into light sleep, or when they're sleep deprived. And so if you consider, say, a seed-based connectivity analysis and place a seed region in an area like posterior cingulate cortex, which is a part of the default mode network, and you look at its correlations with every other voxel in the brain, then you're more likely to see more widespread correlations across the cortex when somebody is drowsier. And this particular illustration is from Tom Liu's group at UCSD. They were doing these caffeine studies, and they scanned people before and after giving them a caffeine pill. And so you can see that this more global changes are happening before they take caffeine. So if you're wondering what's happening to your bold signal after you have coffee, then you can now have some kind of visual. So this figure also presented in their paper, they also had compared, what happens with global signal regression. So if you've discussed that a little bit, I've heard. So we might say, well, if we want to kind of make these more similar to each other, then perhaps we can just, since this arousal effect seems to be quite global, what if we just take our average time course and regress it out of the brain? What happens? So indeed, it does reduce the correlations when you apply global signal regression. But you still see some, if you apply global signal regression uniformly to all your scans, maybe it's starting to remove some of the common arousal fluctuations in in this kind of scan, but in the other one, where the person is already more alert, you might start to just kind of remove other things as well, because the global mean signal is kind of just folds in the average of whatever is there. And if you don't have a kind of common effect across the brain, then it's going to just take signals from networks and so forth, and could actually artificially inflate negative correlations in the brain. Oops. Okay, so but you don't always just need something like caffeine to see this kind of effect on functional connectivity. If you just have somebody in a resting state scan for 10 minutes and they're closing their eyes, if you actually, instead of calculating one measure of, or one seed-based correlation map across the entire scan, if you were instead to kind of look over the beginning of the scan, maybe take some chunks of time from the beginning to the middle to the end and make separate seed-based correlation maps, you can also see differences in the appearance of these maps, which will actually be the subject of the next lecture. But one thing we wondered is, even in the course of a single scan, do changes in the connectivity patterns reflect to some extent the arousal state of the subject? And so to study that a bit more, we had subjects wearing an EEG cap. So EEG measures electrical potentials on the scalp, and there are some clear signatures of alertness, which we'll discuss a little more. And so based on the power in the EEG signal in certain frequency bands, within these different windows of time across our scan, we could actually try to label them as being more alert or more drowsy, and then see what kinds of fMRI patterns corresponded to those changes. And so overall, we found that when the EEG signal indicated that subjects were more likely to be alert, then you also saw more negative correlations in the brain and less strong positive correlations. Conversely, when they were likely to be more drowsy or falling asleep, then you saw more positive correlations between cortical regions. And the networks we studied in this work were default mode network and dorsal attention network. So along with the changes in those specific networks and seeds, I just wanted to illustrate that if you look more broadly at patterns of network-to-network functional connectivity, then you can also identify these kinds of patterns that may be changing in relation to arousal. And so here they did a clustering analysis. Again, we're going to cover the details of this kind of analysis more in the next lecture, but they essentially made a series of connectivity matrices between networks, and then clustered them to understand the kind of major patterns that arose from the data. And for each one of these, they also call these states, but let's just call them connectivity patterns for now. So for each one of these patterns, they could see how prevalent it was, like at the beginning or the end of the scan. And so like this pattern, for example, might be associated with drowsiness, as it tends to increase its expression over time as the scan goes on. They also had EEG collected in the study so that they could validate, this is maybe likely a drowsy state. But again, you see more correlation, and it tends to be more in the sensory systems that seem to correlate to each other. One note about this work is a kind of a subtle point, but because they used ICA to isolate networks, and because they had some kind of normalization step earlier on, there's less global effects overall than if you were to just kind of correlate signals from the raw data, but you could still see this, this effect starting to come through. So those were just some general observations of effects in fMRI that tend to be observed as people may become drowsy. And so I think, if you see some of these things happening in your data, it might not always be because of this effect, but it's worth maybe asking whether there could be some systematic difference that's worth kind of, tracking down. Maybe one group is more sleepy than another. So in this next part, I'm going to go a little bit more into how you can measure arousal levels in the scanner, and also more about how we can more specifically identify patterns of fMRI signal change that are associated with transient changes in arousal levels. And so there are a number of different ways in which you could record some kind of index of someone's state during the scan. If you have an experiment where your subjects can keep their eyes open, you can monitor the behavior of their eyes, and this is really revealing, actually. You can see when they're starting to close their eyelids more and more. That is a really powerful measure, although it can be confounded by people, deciding voluntarily they're going to close their eyes, so that's just something to be aware of as well. The diameter of the pupil is a physiological measure associated with arousal state. So if you have an MRI compatible eye tracker, you can extract this pupil measures. And then, as I mentioned before, electrophysiological signatures can indicate whether a person may be more alert, or and allow us to do sleep staging when people are kind of allowed to sleep in the scanner for longer periods of time. So there are different frequencies of variation that happen in the EEG signal. One of them is an alpha band oscillation. So alpha is around 10 hertz approximately, and is prominent in relaxed wakefulness. So it's actually really fun if you have an EEG cap on a subject and you just say close your eyes and relax. You can start to see these little sine waves emitted. It's like I find it really cool every time I do an EEG study. So when people are relaxed and they're awake, you tend to see very prominent alpha, and as they become more drowsy, you see an increased prominence of lower frequency oscillations in the data. when I was first starting out and saying, like, okay, what should my quantitative arousal measure be in my studies, I look in the literature, and there's actually so many different electrophysiological measures that have been used in the literature to give some kind of quantitative measure of arousal. And could be a little overwhelming, but there's some pattern to this. In fact, most of these measures take some I would say alpha or beta, which is a little higher frequency than alpha, this kind of general frequency range, and may take a ratio between that and the lower frequencies as an index of alertness. So just a note on this, some measures are easier to acquire in the scanner than others. If you have your subjects closing their eyes the whole time, then the eyelid index is not as relevant. So that's where the electrophysiological signals can be really helpful. And also, aside from just studying vigilance itself, and I think you're going to have a more a detailed lecture on this later, but there's also a lot of other interesting electrical phenomena that you can see in the EEG and then use fMRI to look further at the whole brain correlates of what's going on. But it is tricky to get good data in the scanner. Firstly, you have to have specialized EEG acquisition hardware that can operate safely in the scanner. And then, you still have to deal with large artifacts that are induced in your EEG signals from the fact that you're in a high field magnet and that you're switching your gradients to create your MRI images. And so if you actually look at raw EEG data that's recorded in the scanner, you just see this like black wall of artifact, like nothing. But if you have some kind of real-time artifact reduction happening, then, or if you post-process it, then you can see through the artifacts. And actually, one nice thing about the MRI artifact is it's so repetitive. Like for every TR, you're switching the gradients the same way, so you can essentially build up a template, subtract that out of the data, and it does a really good first-pass job of cleaning it up. This is some cleaned EEG data that was gathered in the scanner, and I just wanted to illustrate some of the spectral phenomena I mentioned. So this is time, this is the spectral content of EEG over the occipital cortex. And you can see that at the beginning of the scan, maybe the subject is in relaxed wakefulness, and you see more power in this around 10 hertz alpha band. And then starts to drop out a little bit, and then you eventually see more and more lower frequency activity in EEG. So if you were to calculate one of the measures in this table by taking, say, the ratio of the alpha band to a theta or theta and delta band, then you get something like this, which is a fairly good proxy of their brain state over the scan. And so in the work I showed earlier, we didn't really dissect the exact signatures of what's going on in fMRI as you have these moment-to-moment changes. We kind of looked at overall trends. You see some amplitude change, you see some connectivity change, but now that we have these quantitative indicators and we can compute them on a time-resolved basis, then we can imagine taking these variations and saying, what happens if we use this as a regressor in our analysis, essentially? What if we capture this? Maybe we convolve it with an HRF, or maybe we look at cross-correlations with the fMRI. What kind of patterns that, in the fMRI data over space at every voxel are tracking these fluctuations in an alertness index? And so it was fun to put these slides together because I try to go to different literature and see how similar are the patterns everybody is finding. And actually there is quite a lot of similarity in what people arrive at using these different measures of arousal and correlating that with fMRI signals. And so this study had used the EEG alpha power fluctuations, and this is an eyes closed scan. And so the blue color of this map actually indicates that many of these cortical regions have a negative correlation with alpha. So as alpha power increases, you're actually seeing a decrease in bold signal. This was a study that we did in macaque monkeys. Long story, but we were looking at data from monkeys, and they had behavioral eye measure of being recorded during the scans. And they really would get sleepy during the scans, and you could see them just really start to close their eyes. And so if you traced out the time course of this spontaneous eye closure, you also saw that it was negatively correlated with the signals from many of these cortical regions. Same with pupil diameter. Now, an exception is there are some subcortical regions that show an opposite sign. So the thalamus is one that comes up in a number of studies as showing an increase in signal when you have an increase in an arousal level. This is just another kind of set of studies. They are also finding similar patterns, but the colors in this slide is these are actually inverted because they were looking at correlates of reduced arousal, whereas it was increased in the other slide. So mentally invert these. But again, you see, this distribution of areas over cortex, a lot of sensory and motor regions involved, and inverse correlations in the thalamus. And this one looked in, as in even deeper. I would say, they're falling asleep here. And you can also see signals in the ventricles and other non-brain tissue, which is also start to become more prominent. So basically, across these collection of studies, there is some convergence on the fact that there may be particular activation patterns in the brain that are linked to these fluctuations in arousal. So we did a study following up on this where we wondered, if these patterns in the fMRI data are reliably linked with arousal fluctuation, then can we just, if we didn't have an eye camera or if we didn't have EEG, could you actually just take the fMRI signal itself and say, I can recognize these patterns, I can find out when somebody is falling asleep? So that was actually the goal of the study. We wondered, can we leverage this knowledge that there may be some pattern happening in order to detect somebody's level of alertness throughout the scan just from the fMRI data itself? And so the idea was you first learn the pattern, and we could kind of adopt it from the literature, but it's useful to just use your own data, use EEG together with fMRI or eye-related measures, and just derive this from a set of subjects. We can call that our training set. So we're using that to build our model of a voxelized pattern that contains the extent to which an area is modulated by correlated with an index of arousal. And in this case, we used alpha to theta power ratio. And then once you have your map, you can go to a separate set of data and say, okay, well, if I don't have EEG or eye behavioral measures, can I actually predict the level of alertness that someone's in, essentially a 1D time course, capturing that at any moment in time or in every TR of our fMRI data. So that produces this red time course, which we call the fMRI alertness index. So that's our estimate based on this fMRI pattern of how alert someone is over time. And because we wanted to see if that indeed matched some true or more validated measure of alertness, then we compared it to the EEG alpha theta power in the scan to find that it matched fairly well. And so the method that we use here is actually a pretty simple linear approach. So if we consider this to be some kind of a template pattern, it's just a 3D map, then in your new scan, for each successive 3D volume that you're acquiring in your fMRI scan, you can actually do a spatial projection of this template pattern onto these volumes. Each projection will produce a scalar value, so that gives you this time course that we took as this estimate. So we first looked at this actually in the monkey data set, where we had eye opening and closing as a putative alertness measure. And so, we try to just derive a time course ourselves using this algorithm. and could find that we could pick out times where the monkey likely had their eyes open. And then just to be sure, this wasn't something specifically linked with the monkey's eye behavior opening and closing. We also just looked at an electrophysiological signal similar to what I introduced in the humans, and found that, you know, even when the monkey has his eyes closed for longer stretches of time, then you can still see that this measure correlated. Just as well as during the periods of time where they were kind of alternating between eye open and closed. So we wanted to just look a little more at this in terms of, you know, how useful this might be in predicting subjects' behavior in a scan. So if you run fMRI experiments before, you might have observed that even if you give same stimuli over time, you can see variability in how a subject responds to it. So sometimes people will respond quickly and then, I don't know, maybe they're falling asleep. It's our hypothesis that they'll start to respond more slowly, or they'll start to even miss the stimuli altogether. And so we actually looked at, we presented a really boring task just as proof of concept to see how much variability we could get. So subject has their eyes closed in the dark scanner tube, and they hear a tone every so often, not that often, so that gives them a lot of time to maybe fall asleep in between, but we had tones presented maybe every 30 to 40 seconds or so. And what we wondered is, you know, there's some variability that's that you see in their behavior. So we wondered that, you know, could we actually, if we look at a window of time before a stimulus comes on, could we actually predict if they're going to respond or not? Like, is our measure of brain state good enough to kind of predict what they are going to do? And so we took a little window of time before each stimulus and calculated our measure based on the fMRI data and tried to understand whether we could see any association. So I highlighted a few scan parameters here, but I think mainly the relevant one right now is we collected fMRI. We also acquired EEG for further validation, as well as respiratory and cardiac data, which actually talk about a little more, but they closely relate to people's arousal state as well. So I'll just highlight a couple of the results. And this work was done by a PhD student, Sarah Goodale, in our lab. And if we just look at trials where a person made a response versus when they didn't, then we could see a differentiation in the predicted level of alertness based on that fMRI pattern recognition method in these hits trials compared to the misses. And we can also look in more detail at the time around every stimulus. So if you take the time when the tone is presented, and we'll call that time 0, then you could see that if you separately average over trials where subjects responded to the stimuli versus when they did not, you could see that there is a divergence in those curves. And we also just looked at whether or not it could also differentiate fast versus slow responses, which it could do that to some extent as well. So one other question we asked in this work was, how much can we shrink down this template pattern and still be able to predict somebody's behavior? So those patterns that I was showing that tend to correlate with these arousal measures, they're very widespread across the brain. And the pattern that we used in the results I just shared were kind of whole brain multivariate patterns. So every voxel has a weight. But just out of curiosity, we said, well, how many voxels do we really need? We probably could get away with quite a bit fewer if there are correlations between those voxel signals. And so that's essentially what we experimented with here. So we pared down the template to get only 1% or so of the voxels, and found that it could still maintain the prediction accuracy fairly well. But what was key is that we retain the voxels that had those opposing signal changes. So we may need just a few representative blue regions, say the visual cortex, which had very strong weights. But we also, the anti-correlated behavior with other areas in the brain tended to be valuable for this prediction. And in this data, because we didn't regress out any effects of white matter or CSF, then we actually saw that some of the strongest effects were in the ventricles. So there's a probably physiologic mechanism as well contributing to these patterns. So yeah, actually there is a lot of interesting work going on in terms of like potential non-neuronal physiological effects that correlate with brain state as well. And Laura Lewis's lab here has been finding that, like, when people are sleepy, you see an increase in these anti-correlated dynamics between global gray matter and CSF signals when people are falling asleep, which might have a potential role in lymphatic clearance. And so in this study by Javier Gonzalez-Castillo, he actually took the ventricle signal and said, like, is this a useful marker of someone's arousal level as well? So if you look at the power of the signal fluctuation from a ventricle, you can also do a fairly good job of predicting, drowsiness over time. So I will summarize some of those main points so far. One is that these fluctuations in arousal are linked with changes in fMRI signals and in connectivity patterns. And there might be quite reliable patterns that we can identify, which allows us to do things like try to estimate arousal levels from fMRI alone, and also try to model these effects to capture task-related behavioral variability. And one practical reason that we thought that these kinds of directions may be useful is if you, say, go to OpenNeuro and download a data set to analyze, most likely it doesn't have any measure of the behavioral arousal or some EEG signal acquired along with it. And so if we can start to infer these kinds of state changes from the data, then it might enrich our analysis or our understanding or ability to even separate the data into people who are sleeping versus those who are not. The measure we have just discussed is also still not like a perfect measure. There are definitely limitations. So the direction of trying to derive better quantitative measures of arousal from fMRI is also ongoing. Any questions about anything? Yeah.
Thank you so much. So I was just wondering, then, if someone just was measuring, say, resting state fMRI, not necessarily researching the arousal subject, would it be, and say they had that measure, would it be, I guess, advisable to just remove anything related to kind of the arousal signal, or maybe they're interested in another country, or maybe somehow indirectly arousal could be related to the type?
Yeah, that's a great question. I mean, it really, I think, depends on the question of interest. So sometimes if you're not interested in the state change, then trying to model that in your analysis potentially as a regressor could be helpful for trying to isolate other effects that are going on. But it also is complicated because as you alluded to, there is some interaction between, say, cognition and someone's arousal state. So even if we regress it out of our signal, it might not, you can't necessarily like get rid of the effect having happened in the first place. It's not quite reversible, but it may still highlight, other changes in the brain that are happening. Another thing you can do if you're concerned about it is to, if there's a way of sort of maybe matching levels across participants. If it's a little bit challenging to get perfect quantitative measures, but I think that's one direction if you at least know roughly if somebody's drowsy or not. And so thanks for asking. That's a really great question.
Particularly because I remember, I don't know much about it, but I remember I thought there was some looking at the effect of what they call fidgeting and resting state data. So I was trying to put this in that context. It's a bit different, but I don't know if you have any comments on that.
I'm not. That's also really interesting, and there's all these kinds of covert behaviors that are happening, and it's it's unclear the magnitude of effect that those have on studies, but I would not be surprised if you see like the people are doing it more often, that there's some kind of average increase in maybe motor circuits or something that coordinate that. So that's interesting. Yeah. Any more thoughts, or go on to the next section? Yeah, and yeah, please feel free to like interrupt me with questions going forward as well. So the last kind of main topic I wanted to discuss in this lecture is on the relationship with physiological changes in the body, like breathing and heart rate. And actually, in starting to study this area of arousal states, it's been just really clearly highlighted for me how closely connected the brain and body are. This is a really like whole person phenomenon. And I think one nice illustration of this effect I pulled out of a study of microsleeps. So this research group was looking at brief periods where people fall asleep. You might or might not have had some of these in a lecture if you stay up late working on ISMRM abstracts. You just kind of fall asleep a second and you wake up, but there's kind of variable length of time that might happen. And so they measured not only some eye behavior, but also the cardiac and the breathing signal and EEG. And on the onset of these microsleeps, you can actually see a lot of these signals change together. So not only do you see changes in thalamus and gray matter, but you also see changes in the heart rate and the breathing volume and other things happening. And if you consider a resting state scan and you're recording some physiological measure together with fMRI, you can also see changes in the relationships between these signals in the scan. And so this is one subject data I acquired when I was a postdoc. And for this one, I had them keep their eyes open, but I had a camera recording their eye behavior. And so they start out with their eyes open, and they just can't do it anymore. Falling asleep is just like 25 minutes. And so when they're getting sleepier, you start to see larger oscillations in fMRI signal, as mentioned earlier, but you also start to see larger oscillations in physiological signals. This specific measure was we had a pulse PPG photoplethysmogram on the finger, and it shows the heartbeat really clearly, but if you take the envelope of that waveform, it actually tracks some blood volume measure in your finger. So this is a fingertip measure correlated with brain here, and you can see that not only do the characteristics change, but the signals seem to correlate in those waveforms as well. So this was just one person illustration where that was pretty pronounced. But we did a little bit more systematic study later on, and so we also looked at, in addition to this, what's called the pulse wave amplitude from that monitor, we also had respiratory volume and heart rate collected during the scan, and this is a very analogous illustration. We had EEG monitoring their arousal state because they had their eyes closed, and as they become more sleepy, you can see more synchrony between these waveforms. Now we can also look at correlations between physiology and fMRI. And the map I'll show you here is doing that in a time-lagged fashion. So just to break this down a little bit, there's a lot of maps here. Going across the row, these are just different axial slices. Going down here is a function of time lag. And so think about at zero lag, you have the instantaneous correlation between like heart rate and a bold signal, and then you shift them relative to one another. And so then you recompute the map after shifting, and then you like successively shift it again. And so you can essentially see how the spatiotemporal dynamics of the bold signal are evolving, with the change in heart rate, and that's what's shown here. Okay, I want to see the slide. So if these patterns are correlating with a measure like heart rate, we might be like, okay, is this neuronal? How much is this vascular? I think the answer is that the patterns that are associated. And again, you start to see some of these familiar patterns that were correlated with other measures of arousal, probably reflect some mixture of these effects. So we know there's neuronal changes associated with arousal as well, and it seems like not only do the physiological and fluctuations become more correlated with fMRI, but EEG and physiology also become more correlated. So just to kind of illustrate that, we took the lagged correlations between an EEG signal and different physiological measures, and just also saw that they also have some correlation. And so basically, like, this points to this picture where there's, especially as people are becoming more drowsy and there's more modulation of arousal, then you have these joint neuronal and physiological effects and bold effects, and these all kind of go together in certain ways. And so there have been a number of studies that try to pull these pieces together now. So if we take data sets that have fMRI and one or more measure of brain electrical activity, say pupil diameter, heart rate, breathing, skin conductance, all these peripheral signals that you can gather, they seem to have a quite reliable correlation with certain fMRI spatiotemporal patterns. And so, they correlate together in rather structured ways, also with the EEG. So this map shows just the cortical areas, and then we expanded that a little bit to show the kind of inner parts of the brain too. Also emphasizing that a lot of regions of the brain are engaged in these changes. So it's fairly global, and you also see some anti-correlation in some of the subcortical regions as well as ventricles. So one note about this as well is that this forms a fairly large component of the variance in fMRI studies. So this was a first principal component over number of resting state data sets that my collaborator had pulled together. So I think it's quite interesting how there's these kind of unified patterns of brain-body synchrony. Just to go a little bit further, we don't only see that in resting state data, but if you were to try to induce some kind of like change in arousal, you might see it. So if you have a task that wakes them up, you can just see more prevalent like changes in the brain and body altogether, as well as this pattern. So I noticed that in some task scans where people seem to kind of suddenly wake up and start doing the task again, you can kind of see a larger change in the brain and physiological measures, and also spontaneously. So in sleep, you have events like Kate complexes that are thought to involve some autonomic arousal, and you can also see structured patterns of change across fMRI signals, electrophysiology, and peripheral body measures of physiology as well. So let's see. I will just briefly, I think I have, I can go through this. There's enough time. So just to note that, like, it's also an interesting question to ask about mechanisms by which these global changes are arise. So the subcortical brain areas have an important role in regulating arousal, and a lot of, there's nuclei in the brainstem and basal forebrain that have these wide-scale projections across the brain, and thought to be involved in, coordinating some of these brain-wide arousal changes and the observed signal changes that we see. Generally, to test this, the causal role of certain brain regions in fMRI data is really hard. I mean, we can try to mathematically model, like, time delays and other, I guess, signal relationships in order to discern how, like, directionality of circuits. But one approach, kind of more generally, that has been taken in the field is to look to animal models where you're able to perturb directly some brain area and then observe with fMRI what the effect is on the rest of the brain. And so we did this kind of study. I was involved in this work when I was a postdoc, and we targeted this region, the nucleus basalis, in the basal forebrain, which is thought to have important roles in these functions. And a collaborator who is able to do monkey surgeries and able to inject substances in certain brain regions, I don't have these skills, she was able to inactivate reversibly this nucleus basalis region. And then we did fMRI scans after, and this was also in the macaque monkeys. And so the experiments, just to understand further the effects that I'm going to show on the next page, we actually did this inactivation of one hemisphere per experiment. So we would inactivate on one side, do an fMRI scan, and then do the other side, and also do an fMRI scan, and want to see some kind of like laterality effects. And that did come out. So if you actually it did this injection of the nucleus basalis in one hemisphere, then it actually tuned down the global fluctuations on that hemisphere. So if you take a whole brain average signal and ask which signals are contributing most of that, it actually comes from the other hemisphere. And it could kind of reverse that effect if you switch the hemisphere that you inactivate. And so it generally suggests that these regions may play a role in the global fMRI effects that we are seeing that relate to arousal states. And one interesting point I'll show here is that the effects were also state dependent. So if the monkey was more alert and we did this inactivation of nucleus basalis, it didn't really have that much effect. I think because there's not so much global fluctuation to begin with. Remember the caffeine studies, things like that. But when the monkey was really starting to lose their alertness, then you would see more dramatic effects of this signal. So it's suggesting that these variations linked with arousal fluctuation, maybe signal variations, may be suppressed by this effect. Yeah, there is a lot more work that is ongoing in terms of mechanisms. I just wanted to kind of introduce you to one study that we worked on in that space. So to wrap up, I'm just going to show you two clinical applications where we considered arousal effects in some way. So the first one is a study of temporal lobe epilepsy that is work we've been doing together with collaborators in the medical center at Vanderbilt. And one motivation for considering arousal in temporal lobe epilepsy is that my collaborator had found in rodent models that seizures may, some certain kinds of seizures may damage brainstem circuitry that may be crucial for maintaining levels of alertness in people or in someone. So basically, in this work, we're examining brainstem circuits in human subjects using EEG fMRI. But one thing that we found empirically is that the patients seem to be more drowsy than controls, as reflected in both behavioral measures as well as EEG measures. And so we have been trying to see, like, okay, how do we contrast patients versus controls when we have these kinds of effects going on? And so we did try to get quantitative staging of their arousal levels and then try to match them, kind of referring back to the question. So if we look at more in a low arousal state in patients and also in controls, then we could directly compare connectivity patterns, then we end up also still seeing that in epilepsy patients, connectivity from a number of arousal and autonomic nuclei in the brainstem are reduced compared to controls. And then I just finally wanted to highlight that, a lot of this work, or in a lot of studies, we might consider arousal changes to be a confound, but I think, there's so much interesting, there are so many interesting directions that can be taken to study these as markers of interest. And so I think this is really interesting area. And so this is work done by some students in our group who are really interested in neuropsychiatric disorders. They took a large sample of subjects from the Nathan Klein Institute database. These participants have both neuroimaging measures as well as some phenotypic and clinical measures. It's not a patient sample, but if you look at a lot of people in the community, there's going to be some distribution in terms of the anxiety levels or other kinds of measures that you are seeing. And so there's no EG recorded in this data, so we kind of use our bootstrapping, like fMRI-based measure of arousal and found that across subjects, if you estimate some level of drowsiness, it actually does seem to be inversely correlated to their state and trait anxiety. And then we want to go back and try to get individual subject-specific maps of the arousal effect across the fMRI data, and then correlate that to state and trait anxiety measures. And we find that there are actually quite a few regions that come out with significant values, including one in the hippocampus. So we were trying to understand what that means, but the kind of message I wanted to provide here is that we probably can look more deeply at how individuals vary in terms of these arousal signatures across the brain and use that as information rather than just focusing on what's common across people. So yeah, just wrapping up here, there are no natural variations in wakefulness that have changes in fMRI characteristics, and these are brain and body effects. And some of the implications for studies, one, if we model these effects, then we can get more sensitive and interpretable fMRI inferences with less data, because if you are mixing states, then you probably will be reducing your sensitivity as well. But also, this important point that these signatures might provide valuable information about brain function and disease as well. So thank you, and yeah.
Questions? Kind of like a two-part question. So first, I just want to confirm, we kind of there's a global trend of like synchronous for different physiological measurements, including like sleep patterns. So is that the case with like the drowsing state?
Yeah. So it seems that kind of momentary fluctuation in this kind of skin conductance, that correlates with the global fMRI pattern I showed. It seems that a lot of these effects become more pronounced when people are drowsier. But there seems to just be more synchrony, which is interesting.
Yeah, like the follow-up would be like if it was like really kind of counterintuitive, that thing that's been heavily associated with like fMRI signal.
Yeah.
What would be like the potential mechanism to explain that?
Yeah, I don't know if I know what the mechanism, except for that if it is a reflection, if it's a peripheral kind of reflection of your arousal state, which is probably being coordinated by, like brainstem circuits, then you will then see a connection between those as well.